On this slide, we can see a drawing of a gene at the top. So remember what a gene is. It's a piece of DNA, and they're showing us that in blue. Here's a double-stranded piece of DNA. And it contains a region that codes for a protein. That's what they're showing you in yellow. And it contains another region that is regulatory, and that's what they're showing you over here to the left in white and green. Now I want to introduce some terminology here. The location of regulatory code and protein code, or what's called the transcribed region, it's important that we have terminology to describe what lies where. So instead of saying things like, well, the promoter is before the transcribed region, or the protein code is after the promoter, we use the terms upstream and downstream. So we would say that this area is upstream from this area. And that's because transcription only proceeds in one direction. The RNA polymerase enzyme is going to come in and it's going to bind to the DNA at this promoter region. And then it's going to move in this direction and transcribe the information in the protein coding area of the gene. So we say that the enzyme is moving downstream here. All right, so those are two terms that you may hear me use as we move through the rest of the lecture. I also want to point out that in this diagram, the entire regulatory code is promoter. And this is just an example. This is an oversimplified example of a gene. In reality, the regulatory portion of a gene can be much more extensive than this. This is just the most simplified way that we can show a regulatory region. And that means it only has a promoter in it. So on the bottom part of the slide, what we can see is that our double-stranded DNA molecule has been opened up in that regulatory area, in this area that is upstream of the protein coding area. And I think you can just make out sort of this ghostly gray drawing behind the DNA that represents the RNA polymerase enzyme. So the RNA polymerase enzyme comes in and it binds to the promoter. And specifically, it's binding to these two green regions. That's not super important that you know. It's just that I want you to understand that this RNA polymerase is specifically binding to particular areas in the promoter that it recognizes. Okay? The RNA polymerase needs a place to begin. And what the promoter does is it lines that enzyme up so that it is directly upstream from the protein coding region of the gene. Now, this looks a lot like a replication bubble here, where the double-stranded DNA has been opened up. Remember, we have to open up the DNA. We have to split the strands of DNA, but not because we have to copy the DNA here. Instead, it's because we have to transcribe a gene. And this is where transcription can get a little bit complicated. There's only one strand of the DNA that needs to be transcribed. Only one of the strands has the code for the protein. In other words, the bases are in a particular sequence or a particular order that will convey the information for which amino acid belongs where in the protein. So one strand will be transcribed. The other strand won't be transcribed. We call the one that gets transcribed the template strand. That makes sense. The other strand is called the coding strand. And I know that can be a little hard to understand. why would you call it the coding strand if it doesn't have the code in it? We'll try to address that a little bit later in the lecture. So the RNA polymerase enzyme lands just upstream of the protein coding region of the gene. The double-stranded DNA will be opened up, and this enzyme will begin to move downstream, and it's going to build RNA on top of the one template strand of DNA. Up at the top here, now all we can see in the DNA is yellow. We're in the yellow region now. So the regulatory region, the promoter, would be back here somewhere. And now the enzyme is just moving forward. It's just charging forward. The replication bubble, which you might recognize during DNA replication, is really a transcription bubble here. And it's behaving differently. Instead of opening up in both directions, it's only opening up over here. So we're separating the DNA as we move forward, as we move downstream. Behind the enzyme, the DNA is actually closing back together again. So you can see this bottom strand here of blue. This is the template strand. And the top strand is the coding strand. So the template strand is where the enzyme will make the RNA. And they've drawn that for us in red. So where the RNA is being made in this moment is inside the bubble. And they've drawn a little arrow on this end because this is the 3' end of the RNA. And new bases will be added to this 3' end. And the bubble will keep opening this way all the way down to the end of the gene. Meanwhile, behind the bubble, the DNA is going to close up again into a double strand, and that will displace the red RNA off of the template strand, and that's why it's hanging like this. Okay? Now, I had mentioned to you that there's something called a termination sequence at the end of the gene, and this is the part of the gene that tells the RNA polymerase enzyme, essentially, that the gene has been transcribed, that it can stop now. So in this drawing, they're showing us the end of the protein coding region in yellow, and then they've drawn this sort of blue region here. And the enzyme, of course, will just keep transcribing into this blue region, just like it did through the yellow region. But I want you to notice what's happening with the RNA. All the RNA that's been made so far is sort of dangling off the enzyme. But look what's happened here. The RNA has folded back on itself in this region. The termination sequence, or what they have labeled here as the terminator, is a short sequence of DNA that is in a particular order of bases that allows the RNA to be complementary to itself in that region. So as soon as that RNA gets made, it's going to be attracted to itself, and it's going to fold and complementary bind to itself. This is going to finish transcription, and the reason is because this is a bulky piece of RNA, and it doesn't hang as nicely as the rest of the RNA does. It doesn't fit well inside the RNA polymerase enzyme, and it's going to cause the enzyme to let go at this point. So all of the information for building the protein is up here. This is the transcript of that yellow region of DNA, the code to build the protein. And then back here, this big bulky piece that's folded on itself, that's just the termination signal. That's not involved, that won't be involved in the production of the protein. So let's take a look at transcription and translation with some actual bases involved, because it's important that we continually think about this complementary nature of these processes. Up at the top on this slide, you can see a double-stranded molecule of DNA, and they've opened up the DNA in one region. We're going to pretend that this is a gene, and specifically this is the protein coding region of the gene. So this is not including the regulatory region, just the bases that have the code to build the protein. So we have two strands, and the bases are all complementary bound to each other. A to T, T to A, G to C, C to G, all the way across this region of DNA. Now what you see happening is transcription occurring. The two strands have been separated, so we have to imagine that the big RNA polymerase enzyme is sitting in here, marching forward, doing transcription. We're only going to build the new RNA onto one of the DNA strands. That's the template strand. The coding strand just stays up here. As the bubble opens up in this direction, it closes back up over here. And we're going to create this RNA molecule. Excuse me. So take a look at the bases. Where there's a T in the template strand, we're going to place an A in the RNA. Where there's an A in the template strand, there's going to be a U because there are no T's. Where there's a C, there's going to be a G. And over here, where there's a G, there's going to be a C. All right, so we march forward. We get to the end of this protein coding region. We're going to have termination signal over here somewhere. That's not important. This is what's important. This is the mRNA, the messenger RNA, that is going to be used for translation. Now, remember how a cell evaluates this. The ribosome is going to bind to this RNA molecule, this messenger RNA, and it's going to read it in groups of 3 bases. So it's going to read AUG as the first codon, the first group of 3 bases. If you recall, when you go to the genetic code table, AUG is the start codon. So this is how the ribosome knows to begin right here, because there's an AUG. So it will sort of jump on here, and it will place an amino acid at the start codon. That amino acid is methionine, or MET. So that's what they're showing us down here. A tRNA molecule is going to come in, and it's going to help the ribosome place a methionine at that first location, if you will, in the protein. Now the ribosome is going to move over one codon, one group of 3 bases. And now it's going to read AUC. And that's the code for isoleucine, or what they have abbreviated ILE. So the ribosome, the tRNA will bring an isoleucine in, and the ribosome will build a peptide bond between those 2 amino acids. And then the ribosome moves over one more and opens up the next codon, which is UCG, which codes for serine. And it keeps going all the way along that mRNA until it hits a stop codon. And that's the signal for the ribosome to stop building this particular protein. Now I want to say one more thing here, and I'm just going to mention it. It's not something I will test you on. But I wanted to go back to that strange language that I mentioned, where we call one strand of the DNA the template strand, and the other strand gets called the coding strand. The reason it got the name coding strand is this. Take a look at the order of the bases in the coding strand and the order of the bases in the mRNA. They are identical, except for the T and U thing. So it's ATG in the DNA, AUG in the RNA, ATC, AUC, TCG, UCG. In other words, with the exception of the T's and the U's, the code, the sequence of the bases in mRNA matches the coding strand. It's complementary to the template strand, but it actually matches the coding strand, again, with the T to U transition. So that's why they ended up calling that strand the coding strand. It's hard terminology, though. It's confusing. We just have to remember that it's the template strand that is used to build the messenger RNA during transcription.